good afternoon. Thank you for stopping by. So we are presenting our work on the gut microbiota and more importantly, the gut microbial metabolites that could predict and modulate response to immune checkpoint therapy in head and neck cancer. As we all know that the responses to immunotherapy is very low in head and neck cancer, and there is a crucial need to find out which patients would respond and how do you actually modulate that biomarker to improve responses. So in that area, we try to look at the gut microbiota and to emphasize the microbial metabolites, if they could be used as a biomarker. What has been found in this space is that the microbial species that are associated with response, they are really not reproducible between studies. Every study, even in the same cancer type, comes up with a different signature. So that is something called functional redundancy, where people share only 40% of the bacteria, but they share approximately 80% of the microbial metabolites. So what we did was that we took 35 patients who were starting immune checkpoint therapy, and we took their blood and stool samples to look at the metagenomic signature as well as the metabolite signature, and we collected similar samples at 3 months when we were assessing response. And we not only looked at the metagenomics, that means the diversity, the phyla, the species between responders and non-responders, we went a step ahead, and that is where I think science is leading us to, is the microbial metabolites. Not we as host produce metabolites, there are a lot of drugs developing in metabolites, but the microbiota produce metabolites, and there is a host-co-host interaction. So what we showed was that in responders, inosine was decreasing with response, while in non-responders, inosine increased with response. This is the first in human study where we are looking at inosine as a microbial metabolite associated with response. There was a paper published by Mager et al. In Science in mice, where he had shown that inosine was associated with response to checkpoint therapy. And the other metabolite that we saw with adenosine, which is again a crucial microbial metabolite, was associated, a higher adenosine was associated with non-response to therapy. Once we had this study, we actually went ahead and took samples from durable complete responders to validate our findings, and we were able to reproduce the same metabolic results in durable responders. So this is the work, and we are trying to develop pathways and drugs that could act on these microbial metabolism.